Hi friends, I am Sariga Jayadevan, Assistant Professor, Department of Biotechnology, AJK College of Arts and Science. In this session, we will be discussing about Introduction to Mendelian Genetics and deviations from the basic Mendelian laws. John Mendel, who is a scientist who first successfully proved what is the reason behind inheritance. Before Mendel, lot of scientists were thinking about what is the exact reason behind the inheritance pattern or what is happening during the time of inheritance. What is the molecule which is responsible for this inheritance pattern? But none of them succeeded to scientifically prove what is the reason behind. What is actually inheritance? It is nothing but it is the process or it is the condition where we are resembling as that of our parents. Now we know that we are showing some of our characteristics as that of our grandparents or parents. This is what called inheritance. But why this happening so? Or is there any pattern for this? That none of them succeeded previous time. Ancient time itself, people were thinking about that. But none of them succeeded. But Mendel, who is the person who first scientifically gave a proof to this inheritance pattern. That's why he is known as the father of genetics. Now, what he did? He did hundreds and hundreds of, even we can say thousands of hybridization experiments and proved the theory or put forward some theories which can be applicable to both plants and animals. And these theories are known as laws of Mendel. And that is the fundamental theory that we can applicable for all the organisms and which is giving an idea about how inheritance is happening. Now, what he did? He did several hybridization experiments. What is hybridization experiment? It is nothing but it is simply crossing between varieties. Even it is plant or animal, we can say it is a crossing or hybridization experiment. What Mendel did was, he selected a model organism and started several hybridization experiments. He chose Pisum sativum as his model organism. Pisum sativum is nothing but it is the scientific name of our simple garden pea. He did hybridization experiment in Pisum sativum and finally succeeded to prove the inheritance pattern. Now the question is why Pisum sativum? But we can't say completely why this Pisum sativum or why Pisum sativum is the model organism. But we can say one thing, why he succeeded, even some other scientists did the same thing with some other organism, they are failed, but why Mendel succeeded? Then we can see why this. What are the characteristics of Pisum help Mendel to get this result? First characteristics which help Mendel to get the better result or this successes, first is the availability of large number of contrasting characters in Pisum sativum. What is meant by this contrasting character? That is different varieties we can see in Pisum. As we know that, we can see some Pisum which is of tall variety or some with the dwarf varieties, or we can see some pea plant which are producing red flower color or some varieties which are producing white flower color. Even we can see some plant with the wrinkled seed or some with the round seed. So these many different contrasting traits are available in pea plant. So this helps him to study loads of characteristics. Actually, Mendel took 7 contrasting characters for his study. What he took was first height pattern. For that he took 2 contrasting traits like tall and dwarf varieties. For flower color he took 2 traits like red flower color and white flower color. For seed shape he took wrinkled and round seeds. For pod shape, he took inflated and normal pod. For flower position, he took axial and terminal flowers. For seed color, he took green and yellow. In such way, he took 7 contrasting characters and he did his experiments. Now, this is the first trait which helped Mendel to get the correct result. That is the availability of contrasting trait. And the second one is it is easy to cultivate. Pea plant is very easy to cultivate. As we know that we don't need to give any much more care for a pea plant to grow. So that is another thing. So that will help him to get a better result. Third trait which helped Mendel to get better result is it is easy to pollinate. Normally, pea plants are bisexual flowers. Bisexual means both male and female gametes are there in the same plant or same flower. In such plants, it is very easy to do the pollination experiment. Otherwise, it is very difficult. And one more thing, if it is bisexual, and one more advantage for Pisum sativum selection is Pisum sativum is always preferring the normal pollination pattern, normal self-pollination pattern. Self-pollination is nothing but it is the process where pollen grains from the one flower is falling on the stigma of the same flower. So that is the normal pollen grain pollination process, that is simple pollination process. But normally nature preferring cross-pollination. Why? Because by cross-pollination only we can introduce diversity or better fit character in nature. So nature always prefer for this cross-pollination. But even our pea plant is self-pollinating, so it is very easy for us to conduct the pollination experiment. And one more thing with that pea plant is we can easily do that hybridization experiment. Say, but for some other plants, it requires certain external pollinating agents. Like some plants require animals, zoophili, we can call them as zoophilus, or some plants require certain bats for pollination. In that cases, it is very difficult to conduct normal hybridization experiment. But this pea plant is a normal pollinating, a self-pollinating one. And one more advantage with this self-pollinating pea plant is it is not showing any protandry or protogynous characteristics. Protandry means in sunflower, to ensure the process of self-pollination, that nature itself do some things, like in some plants or in some flowers, even androecium matures later or gynoecium matures later, so that normal pollination will never happen. So in this case of pea plant, no such barriers or no such things are there in pea plant. So it is very easy to do that pollination process. So that is the another advantage of this pea plant selection. Now, then third one is large number of progenies can be raised within a short period of time. As we know that from a single pea pod, we will get about 8 to 10 seeds, and each can be grown into a new plant. And likewise, from a whole plant, we can raise about 100 to 200 number of offsprings. So what is the importance of that means? We are approaching this problem in a statistical way. Actually, Mendel was a mathematician, so he approached that in a mathematical way. For a statistical method, we need a large volume of sample size. Then only we can eliminate the chance of getting fail. So, by getting large number of progenies within a short period of time, it is easy to make him to get that result. That is another important criteria or another important characteristics which help Mendel to easily get the result. And the fourth one, which is actually not directly connected to his experiment, but whatever all the character which he was selected was not the linked trait. That later we find that they are all not linked varieties or linked traits. What is meant by linked traits? Linked traits are nothing but the traits which are present or genes which are present on the same chromosomes are called linked genes. If the genes are not linked means they have the ability to segregate a path. But in the case of linked genes, they will always go as such or together. That we will discuss in upcoming videos about linkage and all. But right now we can come to the other point that they are not showing any linkage pattern. So these four advantages helped him to get the better result. Now, what he did? He did several hybridization experiments. How he conducted this thing? What he did was, he selected one plant and make them as the male variety and take another plant and consider that as the female variety. And what he did was, he collected the pollen grains of the male plant and put it on the stigma of the another plant, which was considered as the female plant, and then did the hybridization. But here there is a one doubt, because what we need was, we need the result between crossing of this male and female pair. But we already mentioned that this plant is a bisexual plant. So there is a chance that there may be a chance of self-pollination. To avoid that, what he did was, he removed the male reproductive organs from this female flower, and he called that process as emasculation, or we can say that the process by which we are removing male reproductive organ from a female flower is called emasculation. After emasculation, one more challenge is there. What is it? Nothing but it is normally pollinating a normal pollinating plant. So there may be a chance that pollen grains from some different plant may cause cross-pollination with the stigma of this plant. So this may also happen. To avoid that, what he did was, he covered that plant with the paper bags, and he called that process as packing. So what he did was, he crossed the plant, then he emasculated, before crossing he emasculated the female flower, and then he bagged the flower to get the accurate result. So likewise, he conducted about 25,000 of crossing experiments. Actually, the crosses that he done was, we can divide into 5 types. First is a monohybrid cross, second is a dihybrid cross, then back cross, test cross, and final one is the repeat cross. Then, what is a monohybrid cross? Monohybrid, mono means single. It is nothing but a cross which involving only one pair of contrasting character is known as a monohybrid cross. That means, say for example, if I am considering flower color, then I am considering only the flower color. I am not mentioning or not considering any other trait of the same plant. If I am taking a plant with the red flower color and plant with the white flower color, I am not considering whether that plants are tall or dark, whether that plant bearing green pod or white pod, whether that plant bearing wrinkled seed or normal seed. I am considering only this character. This kind of cross is called a monohybrid cross. In monohybrid cross, what he did was, he selected a homozygous variety of red. Why? How he raised the homozygous variety? By keep on crossing red varieties with the red varieties. What he did was, he crossed the red plant with the red plant. That plant which is producing red flower with the same plant producing red flower, or with the different plant producing red flower, and it is continued till he got only red flower. And he considered that as a homozygous variety, and he crossed with the same kind of white variety that he obtained through this process, and he called that as homozygous white variety. And he crossed these parent, and he named the first parent as P1 and second parent as P2, and he raised the first generation, and he called that first generation as F1 generation or first filial generation. F is the short form of filial. F1 generation. In F1 generation, in this cross, what he got was, in F1 generation, he got all red varieties. In the F2 generation, what he did was, he conducted a selfing experiment or selfing cross of the same one. That is, putting the pollen grains on the stigma of the same flower, and he raised the F2 generation or second generation. He called that as the second filial generation or F2 generation. In F2 generation, what he got was 3 red is to 1 white ratio. That doesn't mean that only 4 plants. The ratio what he got was 3 red is to 1. He noted down it. Next, he conducted a dihybrid cross. Di means 2, where what he did was, he selected 2 traits. Say for example, height and flower color. If we are considering 2 traits, height and flower color, we are not considering any other traits. We are just considering only the 2 traits, that height and flower color. Where what he did was, he selected a tall plant with the red flower and crossed it with a white plant with the dwarf variety, or dwarf plant with the white variety, white flower color. And in F1 generation, he got a tall red plant, all tall red plants. In F2, he got a different ratio, which was 9 is to 3 is to 3 is to 1, where we got 9 tall red, 3 tall white, and 3 dwarf red and 1 dwarf white. So this was the ratio he got in the for mono F2 generation of dihybrid cross. Next he did that back cross. Crossing this F1. F1 is the first generation offspring that we got from a cross between P1 and P2 parent. Crossing this F1 with either of the parent, either of that red parent, that is the first parent, or the second white parent, second parent, or in the case of dihybrid cross, that tall red or that dwarf white. Either of that parent is known as back cross. Crossing F1 with its either of the parent. These are all did for ensuring his result and to get some more accurate data for interpreting the results. Next cross that he did was a test cross. Test cross is actually a type of back cross where what he did was, he crossed that F1 with its recessive parent. Now what is this recessive and dominance? We will come to that concept shortly. So we can now say it is a recessive variety. Okay. So what he did was, he crossed that F1 with its recessive parent. That type of cross he called as test cross. Why it is called test cross? Means through the test cross we can identify the genotype of a unknown variety. What is the genotype? Genotype nothing but the genetic constitution of an organism. So if you are telling a flower color, there should be some genes and some gene format is there. That is known as the genotype. And the physical expression of that trait is known as the phenotype. Okay, so to find the genotype, we are conducting normally this kind of test cross. After this test cross, he did several repeat cross. Repeat cross means nothing but it is to do that ensuring the result. That is, he did several same crosses again and again to repeat the result. That kind of crosses are called a repeat cross. After all these crosses, he tabulated all those data, and finally he put forward 3 theories, which is known as the fundamental theories in genetics. Because of all these things, he is known as the father of genetics. And these fundamental theories can be applicable for both plants and animals. And what are they? First law is known as law of dominance or law of unit factor. This law states that every character is controlled by a unit factor. That means there is something which is controlling every character. If we are telling about a flower color, there should be something which is controlling that flower color. For a height, there is something which is controlling that height. For our hair color, something is controlling our hair color, or hair pattern, something is there which is controlling that pattern or hair pattern. Why that something? Because on that day we were not aware about genes, chromosomes or chromosome patterns or nothing. So he called that as a unit factor. Now we know that unit factors are genes. Next thing he told through this theory is that unit factors are existing as pairs. Pair forms. They are not single. They are existing as pairs. So for flower color, he gave that unit factor name as R, and he given that R and small r, that is capital R and small r are there. They are together contributing or controlling that character flower color. If it is capital R, it will become red, and if it is small r, it becomes white. Now one more thing. If it is existing as pairs, in sometimes for controlling their trait, dissimilar unit factors may present. Say for example, if it is capital R, small r means what will happen? What will be the expression? For that only he gave that answer. If such kind of dissimilar factors are there means only one is able to express, the other is not. And the unit factor which is able to express is known as the dominant unit factor. I already told you that we will discuss it shortly, right? This is the thing. Those factor which is able to express is known as the dominant unit factor. Now come to our previous point. See, we already told that if you are crossing a red and white, F1 will be all become red. In the F2, it is given as 3 red and white. That means when we are crossing red and white, red is dominating. Such characters are known as dominating factors, or the gene or the factor which is responsible for that gene, that R or that red is known as the dominating unit factor. And those which is getting masked is known as the recessive unit factor. And now we know that these unit factors is genes, and that pair form is the alleles or allelomorph. This is the first law. Okay. So first law is also known as the law of dominance or law of unit factor. Now the second law of segregation, or that law is also known as law of purity of gamete. In this law, he states that every character is controlled by unit factor, but at the time of gamete formation, this unit factor may segregate or separate each other. That's because of that gametes only receive either of the unit factor. No gamete with the 2 unit factors can be seen. So unit factors will be segregating. They are separating at the time of gamete formation. So we can say that the gametes are always pure. That's why this law is also known as the purity of gamete. So this is the second law. Then now the third law. The third law is called the law of independent assortment. This law states that after the segregation, unit factor present for a character will segregate or separate each other. After the segregation, they are assorting independently. They can combine with each other. Say for example, if capital T, capital R is crossing, that is tall R red variety is crossing with a dwarf white variety, that is small t, small r. F1 will be capital T, capital R, small r. According to the law of segregation, at the time of gamete formation, these factor pairs should segregate. That means this capital T and small t should segregate. The same way this capital R and small r should segregate. But after the segregation, this is the essence of this law of independent assortment. That is, after the segregation, this capital T can combine with the capital R or small r in equal proportion. It can either combine with the capital R or it can be combined with the small r. The proportion or probability for that is equal. The same way this capital R can combine with the capital T or small t in the same probability. So that after this segregation process, we will get 4 different gamete with the equal probability, that is capital T capital R, capital T small t capital R and small t small r. This is the law that independent assortment states. And he put forward all these laws, and later he published his findings in a local journal, that is journal of which was published from abroad, and he published his findings. But on that time, his publishings or his findings were not get enough notice by the scientific world, and he was very disappointed, and he died. Later, after 100 years, three different scientists from three different regions of the world rediscovered his theory. They did some experiments, and finally they published their paper. On that time, the scientific world discovered some person already discussed all those things, and they again took his paper, and again people were thought about or discussed about his paper, and he became renowned as the father of genetics. Who are all that three scientists? That three scientists were that Carl Korensen, Hugo de Vries and Sherman. These three scientists did these experiments and rediscovered Mendelian concept. Now the question is, why on that time that Mendel's theory were not noticed by the scientific world, or why they are not noticed? This kind of a theory was there. Why it is so? The two main reasons behind that is, first, Mendel is actually a mathematician, so he approached this in a mathematical way. On that time, that practice was very new, because nobody was using that mathematical theories in scientific paper. But right now we know that without biostatistics we can't do any paper or any research. So through the help of biostatistics only we can get a conclusion. But on that time it was very new. So scientists were very unclear about what he is studying. The second important factor is, on that time the scientific world is very busy with the Darwin's evolution theory. So that also posed some other why that his theory is not accepted. The third reason is, on that time the concept of gene or chromosome were not known. So he is telling something about a unit factor and existing as pairs and something like that. So on that time that were not acceptable. Later, after 100 years only, this theories got acceptance. Later we considering this theory as the fundamental theory of genetics. Now, but we know that deviations are everywhere in nature. Even though we are telling about these are the fundamental laws, every plants and animals are following this theory. Or in another way we can say that this can be applicable to all the organisms. But there is one thing, deviations are everywhere in nature. We can see some areas where we can see these laws cannot be applicable. So right now we can discuss about that deviations that from the Mendelian fundamental laws. We can classify these deviations into two types: deviation due to allelic interactions and deviations due to gene interactions. Allele means we already discussed that according to Mendel it is the factor pair, but right now we know that they are alleles. According to Mendel, when dissimilar unit factors are present, only one is able to express and other is not. Right. But in some cases, we can see some intermediate expression. Carl Korensen, the scientist, plant scientist, who did some studies in 4 o'clock plant, Mirabilis jalapa, he finds some interesting factor. When we are crossing red color flower of Mirabilis jalapa, or that 4 o'clock plant, with the white color producing Mirabilis jalapa, he finds some difference. That is, he got purple colored plant. Normally what we are expecting is the red color plants or red color flowers. But what he got was purple color flower. He think about it, and he did certain F2 crosses, that is selfing. In selfing, what he got was 1 red is to 2 purple is to 1 white. Then he thought about that, why it is happening so. Then he concluded that in some cases, when dissimilar unit factors are present means, it is not always the dominant factor expressing and recessive factor is masking, but in some cases, that dominant factor failed to completely show its dominance, or it show its intermediate expression, and that thing is known as incomplete dominance. So this is a deviation from Mendelian factor. What Mendel thought was, when dissimilar unit factors are present, only one is able to express. But what that Carl Corenson thought was, when dissimilar unit factors are present means only one is able to express, other is not expressing completely. Or the dissimilar factors present means one is not completely expressing and the other also showing its expression. Or we can say that in F1 we will be getting a blending expression of these two. That's why he got one red, two purple and one white. Now let us discuss about that its genotypes. See what he did, that Carl Corenson did. He crossed it red with white. That means a capital R with one small r. In F1 he got a capital R small r. On that time the phenotypic expression is the purple. Then he again did that selfing. In selfing, that genotype that he got was one RR, two capital R small r and one small r. And when coming to the phenotypic expression, what he got was one red, two purple. At the time when capital R small r present, instead of getting the red variety, it becomes the purple. That is white also showing its expression with the red. And that blending will give the purple color. So one red is to two purple is to one white. That is the 1:2, 2:1 ratio, or we can simply say that the incomplete dominance. Next is the co-dominance. Co means together. So in some cases, if two dissimilar factors are present means one is able to express and other is not able to express. That is what Mendel thought. But in some cases, both are trying to show its expression. Nobody had the ability to control the other, or nobody had the ability to mask the effect of the other. Both are showing to its expression. In such cases, we will get a blended result of both of them. The best example for that is ABO blood grouping. We know that ABO blood grouping is controlled by the allele IA, IB and IO. IA is coding for A blood group, IB is coding for B blood group and IO for the O blood group. Okay. So if IA means we will get A blood type, blood phenotype. If IAO means also we will get A phenotype. Right. For B blood type, IB is also a B blood type and IO is also a B blood type. Now IO is the O blood type, which is the recessive factor. Now what about AB? How we will get AB? When IA and IB comes together means we will get AB. That is the best example for this codominance. That is, both the alleles are showing its expression. When dissimilar alleles are present, both of them showing their expression, which is known as the codominant. So these are the allelic expression or allelic deviation due to allelic interaction. Now come to the allelic deviation due to gene interactions. Now, till now or at the time of Mendel, what Mendel thought was, every character is controlled by a unit factor. That is, a one character or a factor is there, that factor controlling the character. But there are some traits which are controlled by more than one genes. Not one factor, more than one factor is controlling a particular trait. Such type of trait also showing this kind of Mendelian fashion, but certain deviation from that Mendelian ratios. Such kind of factors or such kind of traits are coming under this deviation due to genetic interactions. Now, let us consider certain traits which are controlled by two genes. If two genes are present means, same as that of the alleles, the genes are also started to show some masking effects or dominance effects. But for every gene, there is two alleles. So altogether 4 alleles are interacting at the same time. So it will give some other new expressions. That first it was observed in that comb pattern comb pattern. In the comb pattern, we can see two prominent comb pattern, that are single and P. So rose pattern, P single pattern. There are different type of comb patterns are there: single, rose, P, or walnut, some other types. So here the scientists accidentally they got this result. Some scientists were working on poultry and they did some crossing experiments where what they did was they crossed some rose comb varieties with the P comb varieties. And in his F1 or in their F1 progenies, they all find a new variety, that is walnut. At first they thought that it is because of either codominance or incomplete dominance, because this may be a blending of these two, and because of that they are getting this walnut, and they thought that, and they conducted some selfing experiments. That is nothing but they crossed one walnut with other walnut variety which was obtained by the same Rossby crossing. What they are expecting in F2 was either 1:2:1 ratio or 3:1 ratio. But what they got was 9:3:3:1. That is 9 walnut, 3 rows, 3 pea and 1 single. That was a surprising result because they consider one trait, that is compactant. What they got in F2 was the dihybrid ratio. Dihybrid means where we should consider two traits. But we consider only the compactant. Why it is happening so? They thought. And finally they concluded that this particular trait compactant is not controlling by a single gene, but it is controlling by two genes. Because of that reason, the two genes and their alleles are interacting. That's why we are getting this kind of a dihybrid ratio. And this kind of interaction is known as co-epistasis. And if two genes are present and two genes are interacting each other and they are giving a result, means such type of interactions are called epistatic interactions. And here also genes may show this kind of dominance inheritance pattern. If the gene showing epistatic interaction, if one gene masking the expression of the other gene, means that gene is called epistatic genes, and the gene which get masked is known as the hypostatic gene. So this is the epistatic interaction. And what we observed in the chicken, that is called the co-epistasis. Why it is co-epistasis means in walnut, where we can see both the alleles are expressing, both the genes are showing its expression. That's why it is called co-epistasis. Now some more type of epistatic interactions are there. They are called dominant epistasis, recessive epistasis, duplicate dominant recessive epistasis and duplicate recessive epistasis. First is recessive epistasis. Recessive epistasis means if a gene in its homozygous recessive genotype masking the expression of other gene, means that type of epistasis is known as recessive epistasis. Say for example, let us consider the coat color in mice. Normally we can see two type of coat color, that is first is black and second is agouti. Agouti means a white color pattern, or let us consider this black color with the normal albino, white color. And if we are crossing that black with albino, we can, as we know that there are two genes are controlling this kind of trait. So let us give A and B for this black color. If that A, capital A, capital A, capital B condition, means that is both the genes are in homozygous dominant condition, means we are getting that black color. And if both are in the recessive condition, means we can consider that as the albino. So capital A, capital A, capital B cross, small a, small b gives a new variety called agouti. Capital A, capital B, small b. If we are crossing that agouti, we will get a new pattern. That is nothing but 9 of them will become black, because that is the dominant one. But what we are expecting is 3 again with the albino or 3 with the yellow, something likewise we are expecting. Why? Because the individual expression of A gene is black and individual expression of B gene is actually yellow. So what we are expecting is in the 9 case, the first 9 case it will be black. But for the next 3, it is actually A dash small b condition. So in that condition, we are expecting again a capital A black variety. And in the second case, small a b dash, where we are expecting, in the first case we are expecting A, that is yellow variety. And in the second case, we are expecting a black variety. In the final, we are expecting a single one. That is what we are expecting. But what we are normally, or what we are originally got is 9-1 become black, that is A dash B dash become black. But A dash small b, instead of it become yellow, it become a albino. Why this so means yellow variety is not acceptable by nature, or not, nature will never prefer a yellow variety. Why? Because a mice is a nocturnal. It is a nocturnal animal. So if it is come out with a yellow color means it is easy for predation. So nature will not accept or will not prefer that kind of a variety. So it become albino. And again small a, small b become albino. Now the ratio is drastically changes from 9:3:3:1 to 9:3:4. It is called recessive epistasis. Now dominant epistasis. That is another scenario. If a gene in a dominant condition, any allele of that gene is in dominant condition, it will mask the expression of all other genes. The better example for this is flower coloring, that gourd varieties. Mainly that summer squash. In summer squash, there are two genes which are controlling flower color. They are W and A. And the interesting fact is that if W is present in a dominant condition, whatever is the other gene, it is always become white. And the green, that A gene is expressing only when there is W is in the recessive condition. So if you are conducting a cross with a capital W, capital A, with a small w, small a, in the first generation we will get capital W, capital A, small a, which obviously become the white one. If you are conducting a selfing, that is capital W, capital A, small a, with a capital W, capital A, small a, 9 of them with the capital W dash, capital A dash genotype, that obviously become white because of the presence of that W, capital W allele. In the 3, it will be capital W dash, small a, that also will become white because of the presence of that dominant allele. And again the 3, which is of small w, capital A dash, where the A can show its expression because there is no capital W allele. So in that condition only A can show its expression, and on that time it become green or the peach color, whatever it is. And in the last case also, the small w, small a condition, where also that small a can express its, show its expression. Why? Because the W gene is not there for masking its expression. Now that ratio changes from 9:3:3:1 to 12:3:1. This is the deviation. Next is the duplicate dominant epistasis and duplicate recessive epistasis. In duplicate dominant epistasis, where we can see in every pathways there is an involvement of some genes and some pathways. Say for example, consider the flower color producing pathway. Suppose A is the gene which is converting to B with the help of a gene X. Gene X, help of gene X means gene X is producing an enzyme through a transcription and translation, and which is producing an enzyme. A is converting to B with that enzyme, and B is converting to C, that is the final product, with an enzyme Y, that is or the enzyme which is produced from the gene Y. If X is present and Y is absent, the final product won't happen. If X is not there, Y is there, final product won't happen or final product won't get. If X and Y are not present, we won't get the final product. We will get the final product only when X and Y is present. This kind of condition is known as recessive epistasis. That is, in the first case is the duplicate recessive epistasis. In the first case, that is in the recessive epistasis, what we discussed was if one gene in its homozygous recessive condition masking the expression of the other gene. But here the scenario is different. Both the gene has got the same recessive epistatic characteristics or capacity. So that if first gene is in recessive condition, homozygous recessive condition, which will mask the expression of the second gene. The same way, if the second gene is in homozygous recessive condition, means it will mask the expression of the first gene. This is called duplicate recessive epistasis. Then what will be our result? Say for example, let us consider a cross where A and B gene are showing that recessive epistasis. And we are conducting the cross with the capital A, capital B with the small a, small b. In the F1 generation, the progeny will become capital A, capital B, small b. In F2 generation, obviously what we are expecting is 9 capital A dash, capital B dash, where we will get the result. right? Because both A and B genes are present, we can expect the result. So the 9, we can expect the final result. What about the remaining? In that 3, the genotype what we will be getting is A dash small b small b. On that condition, only the first gene is there, second gene is absent. So no final result. So there is no final result for that 3. The second case also small a b dash. Here the first gene is absent, only the second gene is functional. There also we can't expect the final result. So in that 3 also we can't expect the final result. In the last case, both the genes are absent, only small b, small b condition. Both the genes are absent, where also we can't expect the final result. So what we can say that 9:7 become the ratio. The whole 9 will show the result, but the 7 won't show the result. Or if it is color means, we can say that 9 will show the color and 7 with the white color or beige color or off color. That is the result. This is called the duplicate recessive interaction. The same way we can say another kind of interaction which is called a duplicate dominant interaction, where another kind of pathway is existing. There is also biochemical pathway for controlling flower color or whatever it is for that particular trait, where the pathway is like this. There is a product that is first a substrate A which is converting to a product B. Both the genes X and Y are acting on the same pathway. Either X is present means we will get the result, or Y is present means we will get the result. We will get, if X and Y all there means also we will get the result. We will lack the result only when X and Y are absent. This is called a duplicate dominant AJKCAS. Then the ratio will become what? 15:1. The ratio will become 15:1. This kind of interaction we can see in either flower color in some other indigenous varieties of the plants, where we can see this kind of particular interactions. These are the main deviations that we can observe from the Mendelian ratios. Now some other tips for this Mendelian ratios. We discussed about Mendelian first law, second law and third law. What was the first law? First law is stating that all characters, every character is controlled by a unit factor, and these unit factors are existing as pairs, right? And some unit factors, if dissimilar unit factor present means one is able to express and other is not expressing, dominance, recessive and something else. But from where he concluded this result? Through his monohybrid cross, he concluded all this result, right? So, in other words, we can say that a monohybrid cross is the base for a first law of Mendelian genetics. Now, come to the next law, that is law of segregation. What it states? It states that at the time of gamete formation, factor pair present for a character will segregate or separate each other. This also can be explained through a monohybrid cross, isn't it? We can explain this law also with this monohybrid cross, or we can say that Mendelian first and second law can be explained by using a monohybrid cross. Now, what about the third law? What was the third law? The third law stating that after the segregation, the factor pairs are assorting independently. For showing that assortment, we need what? We need a dihybrid cross. Or else we can say that through a dihybrid cross only we can explain the law of independent assortment or third law. Now come to the molecular genetics. What is the basis for all these things? As we know that the gametes are formed at the time of meiosis. Meiosis is the cross which is responsible for this gamete formation. Now, at what stage of meiosis does this segregation is taking place? At what phase of this meiosis does this independent assortment is happening? The molecular evidence is showing that at the time of anaphase is responsible for this segregation. If the genes are linked, or if the characters which we are studying is present on the same chromosome, they are segregated together. And if our gene of interest are on the same chromosome, their separation is taking place in the anaphase stage of the meiosis. And what about the independent assortment? The independent assortment is made possible only because of the alternate arrangement of chromosome at the meiosis phase. So meiosis phase, the meiosis where the genes During that metaphase, the chromosome become aligned on the equatorial plane of the cell. During that time, that alternate arrangement that cells maintain. This alternate arrangement is actually responsible for this independent assortment. And one more interesting fact is that this independent assortment is actually contributing this much difference in siblings. We know that siblings born from the same parent, even though they are from the same parent, they are showing millions and billions of differences. Why it is happening so? Since we are from the same parent, sometimes we may think about that me and my sibling is from the same parent. Why we two are this much different? This is the main reason behind this is because of this independent assortment. As we know that when a gamete is producing at the time from a 23 chromosome, the 23 chromosomes are segregating, 46 chromosomes, they are the 23 pairs, they are segregating and going to each gametes and forming a gamete. So the chance of getting the same gamete is 2 raised to 23 times from the first parent. The same way for happening the same gamete in a lifetime is 2 raised to 23 times only. So altogether, for producing a same offspring or a likewise or same type of a baby, second time is 2 raised to 46 times. 1 by 2 raised to 46 times the probability. That's why we will not having a same sibling, identical sibling. Identical twins are there, but identical siblings are not there. So these are all about the introduction to Mendelian genetics and deviation. In upcoming videos, we can discuss about the molecular structure, molecular nature about these genes and all. Thank you.